Hello, my name is Roy. I'm in Jay Lennon's lab at Indiana University, and we studied the minimal cell in the context of evolution. Because unlike pretty much any other engineering discipline, synthetic biology makes products that are not stable and permanent and unchanging, but because they are imperfectly self-replicating entities, they are subject to the forces of evolution. And in particular, we this can be this can be a bad thing, this can be a good thing, right? If you're an engineer, you want to make something, you want to know what you're making, and the fact that it can change via mutation and natural selection may be disconcerting. But at the same time, this can be we can use natural selection to our advantage, perhaps. If you are designing an organism to perform a certain function, you can use natural selection to improve it. And we were interested in the minimal cell as an evolutionary entity, specifically because, unlike every other organism on the planet, it only has essential genes. And this could constrain the evolution of we use Syn3b, because one thing that we know from evolutionary biology is that essential genes evolve more slowly. So we wondered whether the minimal cell might be constrained in its evolution compared to, say, Syn 1.0. And we also wanted to test whether mutation was changed in the minimal cell compared to Syn 1.0. Again, because genes have been removed, and so this, including DNA replication and repair genes, and so this might alter the rate and spectrum of mutations. And then we also wanted to look at the genetic mechanisms that underlie what adaptive evolution should it occur in the minimal cell. And we started by focusing on the mutation problem, because if mutation rates are different between a minimal cell and a non-minimal cell like Syn 1.0, that could influence the that would influence the genetic variation upon which natural selection could act during adaptive evolution of the minimal cell. So we measured mutation rate using a mutation accumulation experiment, which is performed by repeatedly bottlenecking populations of cells. So they're streaked out on Petri plates, and then we pick colonies at random so that rather than natural selection choosing the variants that arise, the variants are chosen at random by the researcher. And we made sure to randomize, of course, by placing a dot on our plate so that we were picking before we even knew where colonies were. So serial bottlenecking reduces the influence of natural selection. Evolution proceeds essentially neutrally, and of course, as Kimura showed, when evolution is neutral, you can measure the mutation rate just by measuring the observed rate of fixed mutations at the end. So that's exactly what we did. We did whole genome sequencing at the end. And there were two main observations about the mutation rate in the minimal cell. First of all, just looking at the mutation rate of the non-minimal Syn 1.0 and the minimal Syn 3 together, Mycoplasma has an extremely high mutation rate, higher than any other bacterium that has been measured. And this makes sense to some extent, given what we know about Mycoplasma. It doesn't have mismatch repair, and there's features of its biology that suggest it has a low population size, which which is often associated with higher mutation rates. The other really important finding here is that mutation rate was not altered by genome minimization. So the minimal cell did not have a higher or lower mutation rate than the non-minimal, which is interesting on its own, but also suggests that when we proceeded to look at a natural selection context, any differences we observed in the way that the minimal cell evolves would be attributed to differences in natural selection per se and the way it acts upon an extremely streamlined genome. So that's what we looked at next. We asked whether evolution might be constrained, adaptive evolution might be constrained in the minimal cell, given that it only has essential genes. So this experimental setup is similar in that we're doing repeated transfers of bacteria in order for evolutionary time to occur, but we're using liquid cultures here. So populations are large, so that when beneficial variants arise, natural selection promotes them, and we can observe more naturalistic evolution happen in action. And so we did these transfers for 300 days, which is about 2,000 generations of evolution. And then we measured the evolutionary fitness of both non-minimal and minimal cells. And we measured this evolutionary fitness by doing direct competition against the M. cherry labeled mycoplasma. So basically, we were able to ask, how well do your evolved bacteria grow and survive in stationary phase over the 24-hour period that is equivalent to 1 day in this evolution experiment that lasted for 300 days. So by measuring the proportion of evolved cells compared to marked cells at the start of that period, and then at the end of that period, we can ask, how did that ratio change? And we can calculate fitness. So we measured fitness on the non-minimal and minimal ancestors and evolved bacteria. And here's what we found. First of all, just paying attention to this left column here, you can see that, as we've already talked about at this workshop, the minimal cell is sick. Its fitness was cut in half compared to the non-minimal SYN 1.0 in blue. But the minimal cell through evolution was able to recover almost all of this lost fitness, 80% or so. And in particular, the rate at which it recovered fitness was the same as the rate at which the non-minimal cell gained fitness. So this suggests that, in fact, the minimal cell is not constrained in the way it can evolve, which is, again, kind of counter to my expectations as an evolutionary biologist, given that we know that essential proteins tend to evolve more slowly. So the next question we asked is, all right, so if the minimal cell evolves at the same rate as the non-minimal, are the genetic mechanisms the same or different? And so we did genome sequencing, and what we did was we looked at all the mutations that were observed in the evolved bacteria, and we made basically a big matrix for each evolved population and each gene across the genome, looking only at the shared genes between these two strains, of course, so looking at the essential genes. And then for each population, for each gene, how many mutations were there, and we decomposed this giant mutation matrix down into 2 dimensions that I've plotted here. And when we decompose this composition of mutations, what we see is that there is a significantly different composition of mutations between the non-minimal and the minimal cell, suggesting that even though they adapted at the same rate, they were doing this in different ways. And we looked at the specific genes that were involved in adaptation to try to, so this is just like a very general compositional analysis. I then simulated placing all of the observed mutations across the genome at random in order to get essentially p-values for each gene to ask whether each gene had significantly more mutations than was expected given the observed mutations, so that if there was an elevated number of mutations in a certain gene for a certain strain, that suggests that positive selection is acting to increase the frequency of mutations in that gene. And so the output of this analysis is like a big ugly gene table. So I'm just going to focus on one of the significant genes that we found, which is the cell division protein FTSZ. And sorry, I've skipped ahead on slides here. So I just want to briefly tell you, FTSZ is one of the non-essential genes that was added back into SYN3A and SYN3B, because without it and several other genes that have been described in recent work, what you get is you get these really weird morphologies that Shige was talking about, for example. And so we thought, well, if FTSZ is being mutated in these evolved bacteria, that might be affecting their morphology. So we were interested in whether there were morphological changes during evolution. So we looked at the cells under a microscope, and we observed these findings, that while the minimal cell did not change in its size during evolution, it looked pretty much the same as the SYN3B ancestor as well as it did after it evolved. The non-minimal SYN 1.0 increased in size by, it almost doubled in size, essentially. And so this was quite interesting. Especially because, again, both strains of bacteria were getting these FTSZ mutations. Typically, specifically, these mutations were nonsense mutations where it's losing the C-terminal domain of the protein. And so given these observations of morphology, we wondered, well, can we show that this is due to these FTSZ mutations? So collaborators at JCVI reconstructed both minimal and non-minimal cell with the ancestral genomic background, but inserting specifically these FDSC nonsense mutations. And so we're trying to isolate the effect of this single mutation. And what we did was with those cells, we again looked at them under the microscope, and this is what we found. The FTSE caused an increase in cell size in the non-minimal SYN 1.0, but actually slightly decreased the size of the minimal cell, suggesting that yes, indeed, these FDSD mutations account for, at least in part, the evolved divergence in cell size for these two strains. One other thing that we did with these FDSD mutants is that we verified that indeed they do increase the fitness of both the minimal and the non-minimal cell. So this mutation is adaptive for both strains, but has essentially an epistatic effect on cell size, where in one genomic background it has one effect, and in the non-minimal genomic background it has a different effect. I'm about out of time. So I just want to acknowledge undergraduates who helped me with this research, Kelsey, Betsy, JD, as well as funding sources. With that, I'd like to leave you with these kind of discussion points, and I thank you for your attention, and would be happy to discuss if you have any discussion points or questions.
Roy, in the chat, Keone Gandel was asking if I should know this, but are the sequences of the adapted strains, have they been posted?
Yes, we have. Yeah, we have them on NCBI.
Great.
Should be looking at the chat too. Yeah, I can post a link to the NCBI project in the chat.
And the paper is available on bioRxiv.
That's right. I can also post a link to the bioRxiv paper. Stop sharing so that I can see everyone.
Anyone else?
John, could I ask a question? This is Prescott Atkinson.
Prescott.
From UAB. This is maybe a stupid question, but it's still something I've been scratching my head about. We published a paper several years ago, a genomic comparison of M. pneumoniae, 15 different strains isolated over a period of 50 years, several, continents, and the sequences were amazingly identical. And I mean, thinking about the high mutation rate, does this mean this organism is essentially perfectly fit and everything, all mutations are essentially going back to its current sequence? Why would the sequences of all these isolates be so similar? I guess is the question.
Are you asking me or asking John? I'm sorry.
I think I'm asking. Roy, why don't you address that?
So let me clarify. So there's. Can you tell me exactly where the isolate? What are the differences between the isolates?
I mean, the sequences, there are two subtypes of M. pneumoniae, but they're isolated from Asia, from Europe, from North America over a period of 50 years, but the sequences were just amazingly similar. We know this organism can adapt, if it has this inherent high mutation rate, like the one you were describing. It's just amazing to me that it's so similar.
You're right that does suggest that there is negative selection, so selection against new mutations, and that is driving down new polymorphisms that arise. And that would happen because the organism is well adapted to its niche. And which one thing that comes to mind is if we're talking about natural isolates, those are bacteria that have lived in that niche for millions, billions of years. Whereas when we're looking at mycoplasma in the lab, it's in a very artificial environment where it probably is not so well fit. So there is a lot more adaptation to do. So that's one thing that comes to mind, that we're studying the bacteria in a pretty artificial environment where it's common for bacteria to be able to be pretty far away from the fitness optimum in a laboratory environment.
Scott, with M. pneumoniae isolates, so there are some patients that have severe disease and some patients that have minimal disease. Have there been any sequencing studies to look at, does the organism adapt differently in a highly pathogenic situation where another that seems to be almost commensal?
I don't know of any. I mean, I think that that there's been sequencing done in outbreaks. The one that I'm thinking about is one that our CDC did here from an outbreak in Colorado, where there were several uh, uh, instances of this mucosal. There's a type of uh, mucosal and skin involvement that sometimes you can see, and they didn't see any pathogen related specific changes that occurred with it. So it seems to be a host dependent thing as far as pathogenicity goes. But I mean, there could be something that hasn't been seen yet, of course.
You know, I could see if the organism is, and M. pneumoniae doesn't do this like something like M. bucali or, you know, things that really are sort of ignored by the human immune system. But if the organism is under immunological pressure versus an organism that is not, I could imagine that one organism might you know, you might see some evolution, but otherwise, you know, here's M. pneumoniae is perfectly adapted for growth in the organ, you know, in humans. You know, an interesting point of discussion that, you know, might be addressed with our organism in goats somewhere. Anyway, Roy, thank you very much. Troy, here's another example of an adaptive laboratory evolution study.